Hello, everyone. Welcome to today's live broadcast, Elemental Analysis After Microwave Digestion of Various Food Products. I'm Laura Bush, the Editorial Director of Spectroscopy, and I'll be your moderator for today's event. We're pleased to bring you this webcast presented by Spectroscopy and sponsored by CEM and Agilent. CEM is a science-based technology company dedicated to bringing new innovative systems to the laboratory marketplace. Their goal is to simplify science by manufacturing automated systems to classic techniques that provide improved speed, reproducibility, and simplicity. Agilent is a leader in life sciences, diagnostics, and applied chemical markets. The company provides laboratories worldwide with instruments, services, consumables, applications, and expertise, enabling customers to gain the insights they seek. Agilent's expertise and trusted collaboration give customers the highest confidence in the company's solutions. I would now like to introduce today's speakers. We are very pleased to be joined today by Elaine Hasty and Jenny Nelson. Elaine Hasty is a pioneer in the fields of microwave digestion and extraction, having been one of the first applications chemists to develop methodologies nearly 40 years ago. Her continued work has been cited worldwide in various publications, articles, and posters. Elaine is an active member of the AAFPCO Methods Forum Group and AOAC, working closely with both organizations as new methods are developed. Dr. Jenny Nelson is an application scientist with Agilent Technologies, specializing in inductively coupled plasma mass spectrometry, inductively coupled plasma optical emission spectroscopy, and microwave plasma atomic emission spectroscopy. Her broad knowledge of atomic spectroscopy has been applied to both AOAC and ASTM through her serving on expert review panels, chairing committees, and volunteering to develop new methods needed by industry. Thank you both for joining us today. Jenny, please get us started.
Thanks, Laura, for that introduction. Hello, everyone. My name is Jenny Nelson, and I'm an application scientist at Agilent Technologies. Today, I'm joined by Elaine Hasty from CEM, and we are going to share with you some data that we generated at our Santa Clara COE lab in California after the samples were digested by Elaine in North Carolina. We titled this talk, Elemental Analysis After Microwave Digestion of Various Food Products by ICP-MS and ICP-MS. So let's get started. So this is the Agilent Atomic Spectroscopy lineup. We start with the lowest cost entry-level instrument, the AA, and we move along the top row to the Flame AA, the Furnace AA, and then the MP AES. On the bottom left-hand corner, you can see that we have the 5800 and the 5900 ICP-OES, the 7800 ICP-MS, the 7900 ICP-MS, and our triple quad, the 8900 ICP triple. In this experiment today, or this study that we're going to talk to you about today, we used two different instruments. We used the 5900 ICP-OES and the 7800 ICP-MS. Since consumers want to be confident that the food they buy is safe to eat, governments and regulatory bodies in many countries have a legal obligation to protect public health in relation to food. In the United States, the Food and Drug Administration, or the FDA, regulates a wide range of foods. It also publishes details that relate to the analytical laboratory methods used by the FDA to help ensure food safety. For example, FDA Elemental Analysis Manual EAM 4.7 is a comprehensive method that describes how to determine 12 elements in food by ICP-MS following microwave-assisted acid decomposition. It also outlines a series of quality control or QC tests to ensure instrument performance and data accuracy. Today, we'll talk to you about the two different FDA EAM methods that we use to generate data for both the studies that we'll share with you. So if you look here, you can see the 4.7 method, and that requires or specifies that you need to use an ICP-MS for that method. The other FDA EAM method that we used was the 4.4. This specifies that you use an ICP-OES to do the metals determination. You can see all of the samples that we used in this study on the table here. Our good friends at CEM went out and bought a diverse group of food products so that we could really test both of these methods. This slide that we are looking at now is really showing you that we had a variety of food products covering the food matrix triangle of percent carbohydrate, percent fat, and percent protein. You can also see where the 3 NIST SRMs are mapped on the food matrix. For this study, we used the NIST meat homogenate, the NIST slurried spinach, and the NIST non-fat milk powder. And you can see them all in place now and covering that wide range of different samples. Here we are showing you the sample flow for both of the methods that we ran on the ICP-OES with 4.4 and the ICP-MS with 4.7. We first start by homogenizing the food, and you can see the picture for the homogenized frozen dinner and the pizza in the little jars. Our friends at CEM then take the food and digest them in their microwave digester. And from here, I'm going to pass it over to Elaine so she can further inform you about the microwave method and the microwave that she used to do this study.
Thank you, Jenny, and thanks to everyone for joining us today. Before we move into the preparation method, I want to tell you just a little bit about CEM. The company was established in 1978, quite literally by 3 guys in a garage. We are the oldest and largest microwave sample prep company in the world, and we're headquartered in Matthews, North Carolina, just outside of Charlotte. We're often asked, what does CEM stand for? Well, here's our 3 guys in the garage. One guy was a chemical engineer, one was an electrical engineer, and one was a mechanical engineer. So they took their 3 disciplines and they formed CEM. The guy in the middle, Dr. Michael Collins, the chemical engineer, is the only surviving founder, and he's still at the helm today. They started out by making a rapid moisture solids analyzer, and that's what you see in the picture there. Well, why not a digestion system? Well, microwave digestion technology didn't exist in 1978. When I came on board in 1984, it was right about the time that CEM began tinkering with the idea of putting samples and acid in a sealed Teflon vessel and heating it up in a microwave to speed up the preparation part of the elemental analysis process. We only had power time control then, so I'll tell you there was a lot of flying by the seat of your pants and sweating bullets when it came to developing methods. But I'm extremely fortunate to have gotten it on the ground floor of what I think was, and I think still is, a truly revolutionary technology. We've come a long way since then, and this is our current campus in Matthews, North Carolina. All of our manufacturing, engineering, service, applications, and sales support is located here. And with over 50,000 instruments sold, you've probably used or consumed a product that has been tested with a CEM instrument. We have a fully staffed applications team that covers all 4 of our divisions. There are 5 chemists in the analytical group which supports the acid digestion applications, and we are ready to assist you with your sample prep needs. All right, back to the sample preparation method. If you look at the FDA method that we followed, the sample preparation decision tree is split into 2 branches. The right branch applies to an autoclave style microwave system, and the left branch applies to a more traditional closed vessel batch system. In our study, we used the Mars 6 microwave digestion system, so this is going to focus on and use the method on the left branch of the method. In a nutshell, the method requires a half gram of dry weight sample or 5 mils of a liquid sample, 8 mils nitric acid, 1 mil peroxide, heated to 200 degrees and held for 15 minutes, add a little HCl at the end, and dilute to 100 grams with water. So that's the method in a nutshell, and we're going to come back and discuss the method more in a little bit in more detail. All right, here again are the samples that were selected for this study. 10 of the samples were purchased from the grocery store, and each of those samples were digested in duplicate. So that's 20 samples. 2 of those samples, the beef jerky and the gummy bears, were fortified with a low-level spike, and a high-level spike, and each of those were digested in duplicate. That's 8 more samples. There were 3 NIST standard reference materials, each digested in duplicate. That's another 6 samples. Duplicate method blanks, 4 additional method blanks, 2 are fortified with a low-level spike and 2 with a high-level spike. So that gives you a total of 40 samples. So if you're a high-throughput lab that gets a variety of different food samples into test, you want to be able to process as many as you can, as quickly as you can. And that includes your QCs, your SRMs, your method blanks, your spikes. And in our study, that accounted for half of the batch. So again, if you want to be able to process as many unknown samples as you can, then you need a high throughput system. So the MARS 6 iWave with a 40-place 75 ml express vessel set does just that. This table shows the elements and the concentrations that were used to fortify the beef jerky and the gummy bear samples. The big 4 elements are there: arsenic, cadmium, mercury, and lead. They're volatiles, including arsenic, selenium, mercury. There's a mixture of nutritive and non-nutritive elements, so this is a good test of the robustness of the method. Some of the samples required some prep work before the prep work. In any sample, you need a homogeneous sample that's representative of the entire thing so that you get a good analysis. So when you're dealing with samples like a pizza or a chicken pot pie, where do you take the sample from? For the pizza, do you sample the pepperoni, the cheese, the crust? Or for your pot pie, how about the chicken, the vegetables, or the crust on the top and the bottom? So what you need really is a blend of the entire sample. So we took the entire pizza, we took the entire pot pie, and then blended them in a food homogenizer, and then took the ½ gram of the blended sample, and that's what you see in the jar there. But be mindful of what type of grinding equipment that you use. Most kitchen-type blenders have stainless steel blades that can potentially contaminate your sample. The FDA method does discuss that, and they recommend a titanium or tungsten carbide blade rather than stainless steel. Some samples can be a little problematic when weighing and transferring into the vessel liner. Weighing is a very important step in the analytical process, as is every step. But if the sample weight in the beginning is incorrect, then the ICP data at the end is totally meaningless. So you want to make sure that you get a complete quantitative transfer of the sample from the balance to the vessel. But here are some potential problems that you can have with particular types of samples. Powders are subject to issues with static. Teflon liners are notorious for building up a static charge on the outside, and I've literally had powdered samples jump off the spatula and stick to the outside of a Teflon liner. So that can be an issue when we're weighing samples such as our non-fat milk powder, getting them totally into the vessel without having it stick to the inside or the outside of the vessel liner. Some of the samples like the meat homogenate or the blended frozen dinner that have a high moisture content, they're really sticky, and that's hard to get them to the bottom. So when you're trying to put them into the liner, they tend to want to stick to the inside of the vessel wall at the top, and you want to make sure that they get down to the bottom so that they're completely covered with the acids and they will fully digest. Other samples like our gummies are a little bit too big to drop the whole thing in there and digest it, so we need a smaller sample size. We need to cut some of that off in order to achieve our ½ gram sample weight. So here are some potential solutions to these problems. Anti-static ionizers work very well for powdered samples. CEM does have an anti-static ionizer, but there's others on the market as well. You can put your Teflon liner in front of the ionizer, and it will discharge the static on the Teflon so that when you're transferring the powder inside the vessel, it'll slide right to the bottom and doesn't stick to the top. For sticky samples, using a Teflon-coated spatula or a plastic scupula works very well. If you're using a Teflon spatula or a metal spatula, you want to make sure that it's Teflon-coated so that you don't contaminate the sample. For samples like our gummies where you need a smaller piece of it, you want to cut some of it off. You want to make sure to use ceramic scissors rather than regular metal scissors, again, to reduce the chance of contaminating your sample. We have more tips and tricks for weighing samples, or tricky ones and problematic ones. On our website, we have a whole series of videos called In the Lab, and a lot of those videos feature my colleague, Macy Harris, who's going to join us at the Q&A session at the end. So I encourage you to go to our website and check out some of these videos. All right, let's look at the digestion equipment that we used in our study. We used the MARS 6 iWave digestion system with 75 ml express vessels. The iWave features a contactless temperature sensor that measures the temperature in all vessels. It's very simple to use. There's no connections to make. So you simply load your samples into the turntable. You place the loaded turntable into the microwave cavity, you close the door, and you press go. So it's very simple to operate. There are sensors on the inside that detect vessel type, they detect the number of vessels, and that simplifies the method creation process. So there's less decisions for the operator to have to make. There are pre-programmed one-touch methods that are ready to use, or you can create custom methods with specific parameters, such as those that we use that are defined in this particular FDA method. The express vessels that we use, the 75 ml express vessel, is a simple 3-part vessel design that's also very quick and easy to assemble. So what we have here is a high throughput system that's easy to operate. Here are the method parameters that we followed as specified in the FDA method. We used a 0.5 gram of our dry samples or 5 gram of the liquid samples. That was our Ensure nutritional beverage sample. To that we added 8 mils of distilled nitric acid and then let the sample sit open and pre-digest for 15 minutes. Whenever you add nitric acid to a food sample, especially those that are high in carbohydrates and sugars, you're going to see an immediate reaction. You might see some foaming or you might see some off-gassing. And that's because these particular components are easy to digest, so they're going to start really digesting right away. So you want to let those initial reactions subside before you move to the next step. So after about 15 minutes, we added 1 ml of 30% hydrogen peroxide and again let the vessels sit open for another 15 minutes, allow all the samples to finish that pre-digestion, and then they were capped and loaded into the turntable. Now this is not the only digestion protocol that you can use for digestion of food samples. In fact, you want to use the minimum number of reagents possible, and that just reduces the potential for contamination from the reagents themselves. We typically don't use hydrogen peroxide when we're digesting samples, but it does boost the oxidation potential. But really with nitric acid, if you get it hot enough, and 200 degrees generally is, you'll get a complete digestion of your sample without the need of peroxide. HCl can be added or left out. It really depends on what you're looking for. It's there to stabilize metals like mercury, iron, aluminum. So again, you can use HCl or not, just depends on what you're looking for. But if you need help in trying to decide what the correct method is for your particular sample type, please feel free to contact our application staff, and then we can help you select the best method for what you have, your type of samples and the analytes that you're looking for. As far as the digestion protocol, the heating method is concerned, the method specifies a 25-minute ramp to 200 degrees and a 15-minute hold. So that's a total heating time of 40 minutes. You add on to that another 20 minutes of cool down time, and in 1 hour you have processed 40 samples start to finish that are now ready to be diluted for analysis. Here's a snapshot of the run in progress. It's about 10 minutes into the ramp, and you notice the batch temperature is at 110 degrees. That 110 degrees represents an average of all 40 of the vessels in the turntable. You can see the temperature of every vessel position as displayed on the bar graph, but the temperatures are quite uneven. Well, why is that? Well, think back to Jenny's food pyramid. Food samples are composed of carbohydrates, proteins, and fats, and these each decompose at different characteristic temperatures. Carbohydrates tend to start digesting at about 140 degrees, proteins a little higher, about 150, and fats even higher at about 160 to 180. And oxidation of organic material is always an exothermic process. So the samples that are highest in carbohydrates and sugars, for example, the milk powder, the rice noodles, the donuts, they're going to start digesting quicker and at lower temperatures than samples that are higher in proteins and fats, like the cheese and the meat homogenate and the beef jerky. So this pattern is very, very typical when you're batching samples of different compositions together. And remember, we have method blanks included in the batch as well, so they have no sample at all in them. So again, the heating rates are going to differ from sample to sample, and that's very, very typical, and it's okay. Fast forward to the end of the method, and we can see that the batch temperature did achieve 200 degrees. And you can also see that the individual vessels in the bar graph have tightened up quite a bit. They're still not perfect, but that's okay, because again, we know that there are different components in different samples, including our method blanks. But the object of any digestion method is to achieve a temperature that's hot enough to get even the coolest samples in the batch to a hot enough temperature to achieve complete oxidation. And with 200 degrees for food samples, we can achieve just that. Upon completion of the heating program, the vessels were allowed to cool to less than 50 degrees as specified in the method, and that took about 20 minutes. The vessels were then vented and opened, and the contents were quantitatively transferred to sample tubes and diluted to about 50 grams with 18 megaohm deionized water. We then added a half a ml of concentrated distilled hydrochloric acid, again as specified in the method, and that's to stabilize mercury. And then the samples were diluted to a final mass of 100 grams with deionized water. And as you can see in the picture, all of our samples were clear, colorless solutions with the exception of one, the powdered donut samples. We noticed upon dilution that the sample looked slightly hazy, and upon standing we started noticing these fine white particles settling out, and we saw that in each of the duplicates. So we asked ourselves, what's different about the powdered donut samples and all the other samples that digested completely at 200 degrees with nitric acid and peroxide? So we started thinking about the ingredients that are in our samples, and we know that often food manufacturers will add inorganic ingredients such as silicon dioxide and titanium dioxide to their food products as anti-caking agents. So we checked the label on our donut bag, and sure enough, there's the titanium dioxide. Not only is it an anti-caking agent, but it also adds texture and brightness to food samples. So if you're making a white powdered donut, it's a very logical ingredient to add. However, for us it's a problem because it's not soluble in nitric, peroxide, or hydrochloric. You need a little HF or dissolution of titanium dioxide. So the question now is, do we add HF or do we not? And the answer is no, because that's not part of the scope of this particular method. So we simply let the particles settle out, or we filter them prior to analysis. So now that our samples have been homogenized, digested, and diluted to volume, they're now ready to be analyzed on the ICP. So I'm going to throw it back over to Jenny.
Thank you, Elaine, for sharing all that information on the microwave digestion portion of this project. I learned a lot from you. So now that you have seen the digested samples and the ICP-MS that we are using here, or we did use in Santa Clara, I will share with you the data that we obtained on the 7800 ICP-MS using the FDA EAM 4.7 method. So this method, the 4.7 EAM method, is validated for the elements shown here, which is chromium, magnesium, nickel, copper, zinc, arsenic, selenium, molybdenum, cadmium, mercury, and lead. This method is for organic matter such as lipids, carbohydrates, and proteins. This method is not validated for dietary supplements like silica or geological foods like coffee creamers or zeolite dietary supplements. This method is also not used for speciation analysis. So if you needed to know the different species in a food sample, like maybe arsenic 3 or arsenic 5, this method would not be able to do that. You would have to do a speciation method, which the FDA does have, like a 4.10 or 4.11 method to determine that. The elements that are also not validated using this method are iron, iodine, magnesium, silver, and calcium. The 4.7 ICP-MS method spells out all of the polyatomic interferences as well as the elemental interferences that one would need to worry about with this method. So this chart was just taken from the 4.7 method. And so the FDA has done all the work so that you know what could potentially be an issue with the food samples that you're analyzing. It is now easier than ever for food testing laboratories using the EAM 4.7 method to carry out the analysis using an Agilent 7800 or 7900 ICP-MS. Both of our single quad ICP-MS systems are equipped with the octapole reaction system generation number 4. The 7800, which we used for this study, uses helium collision cell mode and kinetic energy discrimination, or KED, to control common polyatomic interferences, leading to more accurate results. To break up polyatomic ions with a higher bond energy, enhanced helium mode can be used instead of a reactive gas, such as hydrogen or ammonia. Avoiding reactive gases ensures that no new molecular interferences are formed in the cell, improving the quality of the data and streamlining the method. This instrument has 10 orders of linear dynamic range and also simplifies method setup, as major and trace analytes can be measured in a single run, meaning there's much fewer reruns due to overrange results. The 7800, which we used for this study, features Agilent's high matrix introduction, or HMI technology, as standard. This improves the ICP robustness and enables it to handle samples with a total dissolved, or TDS, content between 0.5 and 3% HMI. HMI uses aerosol dilution to handle high matrix samples, reducing sample preparation time and minimizing the risk of introducing sampling errors or contamination from conventional liquid dilution. So let's talk a little bit more about the helium collision mode, or the KED. So our 4th generation octopole reaction system, which we already said we use in helium mode. So the helium collision mode is used to deal with interferences. In this diagrammatic explanation, if the ions leaving the collision cell have the same mass and charge, but the polyatomic ions have less kinetic energy as a result of more collisions with helium atoms, then the polyatomics can be stopped by applying a positive electrical gradient between the collision cell and the quadrupole. This approach has been hugely successful and used routinely by labs around the world on a daily basis. This slide shows an example calibration setup. We calibrated most elements from 0 to 25 parts per billion. However, the exceptions were mercury, which was in much lower concentrations, and copper, manganese, and zinc, which were calibrated to a higher level. Here's our block diagram of what this method would look like. So you can see we start with our initial calibration with different concentrations for those different elements I just told you about. Then we do an initial QC check, and then we run our samples, our sample duplicates, and then our fortified analytical solutions and those duplicates. And in between every 10 samples, we run a periodic QC. This is the 4.7 data from the validation that the FDA completed. So we wanted to compare and make sure that our data would be equal or better than the limits of detection and quantification data that you see on the screen now. So let's take a look at our data. So you can see here we've listed all the elements on the far, let's get my pointer out here, on the, here we go, on the far left-hand side, and then using the ICP-MS Mass Hunter, we can get the detection limit and the BEC, which BEC stands for background equivalent concentration, and that's just basically an estimate of your background. And then based on the calculations for EMA 4.7, we're able to calculate the LOD and the LOQ based on the method, and we can compare those to the EAM 4.7 nominal analytical limits. And you can see across the board, we performed better than the method. So we knew that our method and our instrument was running very well, and we're ready to run our samples. On this slide, you can see all the different quality controls that is spelled out in the 4.7 method. And for our time today, we will not go through every single one of these quality control checks, but we'll go through the ones at the end of this slide. So you'll have to believe me or read our upcoming publication where we show you more in detail of each of these different criterias. But for our internal standard recovery, we had between 60 and 120%. Our stability check at the mid-level was within 10%. Our calibration curve, we achieved better than 0.995 for our R value. Our ICV verification was within 10%. Our CCB and CCV all performed within specs for the method. And next we'll go Well, here's some data to share with you on the CCV. So as part of the method quality control procedure specified in the method and to ensure the ongoing validity of the calibration, 2 CCV standards were analyzed 5 times during the analytical sequence. As shown in the below figure, all elements reported recoveries within the EAM acceptable criteria range of plus or minus 10% of the actual concentration. And that is shown right here. And this is just a screenshot of our Mass Hunter software. Okay, so to verify the sample digestion process, 2 sets of the 3 NIST SRMs were analyzed in duplicate using the 7800. As shown here in this table, you can see our table coming in. The mean concentrations were in good agreement with the certified concentrations, meeting the QC criteria requirements of the FDA method of plus or minus 20%. And just a reminder, we use the NIST meat homogenate, the NIST non-fat milk powder, and the NIST slurried spinach. Okay, so a check for the reference material. Now let's look at some of the other QC. So next is our Fortified method blank. So to test for the non-spectral interferences or matrix effects, a Fortified method blank, or an FMB, was prepared by spiking the blank at 1 ppb or 50 parts per billion for some of the higher level elements. The Fortified method blank was analyzed periodically throughout the entire method procedure. All recoveries were within the EMA 4.7 method acceptable range of 90 to 110%. And I'll show that in the table right here. So you can see for all the elements that we were looking at, we passed. All right. So on to Fortified analytical portions. So a spike recovery of a Fortified analytical portion test was carried out to check the accuracy of the 7800 ICP-MS method for the food samples. So 2 food samples, which were the beef jerky and the gummy bears, were selected at random. Both samples were spiked with all of the elements at 1 or 50 parts per billion and measured using the ICP-MS. For samples that had naturally occurring elemental concentrations above 5 parts per billion, a 1 ppb spike is reported. For samples with a higher naturally occurring concentration, the 50 parts per billion spike results are reported. The recoveries for all elements in the Fortified food samples were within the acceptable QC criteria of plus or minus 20%, and you can see that on the slide below. Okay, next slide. All right, so we can see that we get a check mark for a Fortified method blank and a check mark for Fortified analytical portions. Now, the last bit of the QC requirements for this method is to take a look at duplicate portions. So this method states that duplicate portions are strongly recommended for every sample, but they're not required for every sample. You can do once every 10 samples. And in this method, in our experiment, we actually did do duplicate portions for every single sample. So on this slide, you can see the quantitative results for our different food samples. I didn't put all the food samples on this table because that would just be too many numbers on one page. But you are seeing the results for 6 of the different food products. In addition to the 12 elements specified in the EMA 4.7 method, we also provided data for aluminum, vanadium, iron, antimony, barium, thallium, thorium, and uranium. So according to IntelliQuant data, we also need to be concerned about doubly charged interferences on some of our elements like arsenic and selenium. And these interferences come from rare earth elements that are doubly charged, like neodymium or samarium for arsenic, or maybe gadolinium for selenium. So since these are food samples, it's not unheard of or not unlikely that this would be a problem. So we used half mass, we measured elements at half mass like arsenic and selenium to get around this issue. Another thing that we wanted to look at this table is to see if any of the elements jump out to us that maybe we want to take a closer look at. And one of those elements that we wanted to take a closer look at, or one of the food products, was dark chocolate. So if you go over to the right-hand column, the far column on the right, you can see that's our dark chocolate. And we were a little bit surprised at the levels of cadmium and lead in the dark chocolate, especially compared to the other foods. But chocolate is known to contain cadmium, and this is present in the soil for where some of the cocoa plantations are. So US doesn't have any limits for cadmium in chocolate, but the European Union introduced maximum levels back in 2019. And that limit for dark chocolate with total dry cocoa solids above 50% is 0.8 milligrams per kilogram, or about 800 parts per billion. The measured concentration value that we got, you can see, was around 208 parts per billion, which is well below the EU maximum level. So that was good to see that we didn't see any food products that had anything over allowable limits. So now we're looking at the duplicate portions, and you can see pretty much across the board, we had relative percent difference of plus or minus 20% for everything. There was one exception, which was the nickel was just a smidge high, but everything overall looked really good. Okay, so next what we wanted to do is talk a little bit more about some of the new features in our software. So when an analyst develops a quantitative method using a preset method, IntelliQuant quick scan data will be acquired automatically. So there's no special startup or separate calibration needed, and we're basically just simplifying the analysis. IntelliQuant automatically acquires full mass spectrum data for up to 78 elements in every single sample with only a 2-second measurement time. So for not adding very much time, you can really get a lot of interesting data. And I can show you where we used this feature when we were doing this method. So again, we have some new IntelliQuant features, and you can see what they are here. So some of them are shown on the bullet point. So we have a TMS, or a total matrix solids, new feature. We also have automatic calibration. Quick scan was renamed to IntelliQuant, and basically this is just giving similar naming to some of our other products like the ICP-OES. And now we have IntelliQuant heat maps, which gives, you can see a periodic table with different colors, and depending on the concentration of the element in the sample, the heat map gets a little bit brighter. Okay, and you can see that periodic table heat map in either the full quant or the IntelliQuant tab. So let's take a little bit closer look at one of these features, and we like to say that this gives us sample insights with this IntelliQuant analysis. And the first one is the TMS, the total matrix solids. So this new function uses IntelliQuant data to calculate the approximate solids level in any sample analyzed. It excludes gases like carbon, phosphorus, sulfur, and halides. It can, like I said just on the previous slide, it can be displayed in both your full quant tabs and your IntelliQuant tab. And TMS relies on correctly calibrated mass response. This is a great diagnostic tool to identify whenever there's an internal standard suppression. And you can see on the right-hand side what the data looks like. So this new tab shows up right beside where you would have your sample name. And you can see here in parts per million, it's giving us the approximate or the calculated TMS for each of the different samples. So now we can take a quick look at some of the TMS data from some of our actual samples. And so if we take a look, you can see that, let me get my pointer out here. You can see that there's a variety of different TMS results for these different food types. You can see that beef jerky is fairly high. It's over 30,000 parts per million. The Fortified nutritional shake is under 4,000. The Gouda cheese is around 15,000. The gummy bears is our lowest, is under 1,000 parts per million TMS. And then we have slightly higher values for the powdered donuts in the dark chocolate. So that's pretty interesting to see. And a lot of people have been pretty interested in looking at the different TMS results in their different food products. So I think you should use this new function and test it out and see if it's adds some value to your analysis. Okay, so there's also very easy setup with the IntelliQuant analysis, and you can do these automatic calibrations. So IntelliQuant has zero setup and completely automatic calibrations. And you can see over here where these new tabs are inserted into our software. So you can see we have the semi-quant background, semi-quant blank, semi-quant internal standard, and semi-quant standard. Okay, and then you can pick which one you want to use for each of the different samples. All right, so more insights that we can get are again using our periodic table map. So again, this is a heat map and it's relative to the highest concentration in the sample. And you can use this in either display it in the concentration or for outliers. And we'll show you some real heat maps for the data that we ran on the food samples in 1 second. Okay, so here's just to show you where those heat maps will show up. You can see that you can either see it in IntelliQuant or in FullQuant, either tab. Another feature that we didn't bring up earlier was now we have this new %RE and %R. So the %RE is percent relative error and %RSE is percent relative standard error. And these are now calculated on the calibration response. And you can see on the in the data analysis pane where that would show up. Okay, and these new features were put in because of the new requirements for some regulated environmental methods. So since sometimes food and environmental worlds sometimes overlap, we wanted to share this new feature with our food friends. And here's just another closer look of where that new %RE and %RSE functions would show up. Also, you can specify in the software what you want to happen on action on failure. So you can see if there's an error, you can choose what will happen if either of those, RE or RSE, are not within the range that you specify. You can ignore and continue. You can say recalibrate, and do a variety of different end action functions. Okay, so let's take a little closer look at two examples of our IntelliQuant data for two of our different food samples. So you can see on the left-hand side, we have our powdered donuts. So you can see the powdered donuts there. Let me get my little pointer. Let's see. So right over here, you can see the powdered donuts. And one element that jumped out to me was titanium. But then if we think about it, we would expect to see titanium because we know that titanium nanoparticles are used in a lot of powdered donuts, especially the ones that are highly preserved and are packaged, not things that are fresh from the bakery. But these powdered donuts that are very, very white, have a very nice white color, and they can sit on the store shelf for a very long time. Okay, so that's probably where that's coming from. And then another thing that we were surprised to see, if we look over here at the periodic table heat map for our dark chocolate, you can see that we were a little shocked to see tungsten. All right, we had tungsten show up as one of the elements in the periodic table. So let's go ahead and use some of this IntelliQuant features to take a closer look at that. So we saw that tungsten show up on the periodic table heat map, and we wanted to take a closer look to see if that was really real. So with the IntelliQuant mass spectrum, we can confirm the identity of tungsten by using an isotopic template to match what would be expected as an isotopic pattern to what was actually observed in the analysis. So as you can see on the screen here, sure enough, we have a tungsten isotope match show up in our analysis for the dark chocolate. So this confirms that we have tungsten present in our dark chocolate samples. So that was a little bit surprising, and we learned something new. Okay, on to the next slide. All right, so we were able to show you that we did the duplicate portions and also complete all of the QC criteria that you need to do for method 4.7. So that was really good. So we were able to complete the method and we're very happy with all our results. So a little bit of insights onto our results. We did see some lead in the dark chocolate, which was higher than the other foods that we looked at. Also, we found some cadmium in our dark chocolate, and we know that that is present in soil samples of some coca plantations, so not too surprising. The U.S. doesn't have a limit for cadmium in chocolate, but the European Union does, and our results were below that of what is the limit in the EU. So that was really good to see. Okay, the FDA also recommends a maximum lead level of 0.1 parts per billion in candy that is likely to be consumed by smaller children. And the measured concentrations in the gummy bears in chocolate were well below the maximum guidance level. So that was good to see as well. So we have a lot more confidence in some of those snack foods that we are consuming. All right. Okay, so now we're going to switch over to our elemental analysis manual method 4.4. And remember, this is the one that we used the ICP-OES for. So again, we used the 5900 ICP-OES for this method. So here you can see we did the same samples, just a different FDA method, and we used the ICP-OES, that new 5900 ICP-OES. Okay, here's kind of the block diagram of how this method goes. You can see we do an initial calibration, which specifies for at least 3 standard calibration solutions for the elements shown here. We do our initial QC check, and then we have our samples as well as that periodic QC that's running every 10 samples. Okay, so you can see that we did the same thing. We have our list of quality controls that we're following for this method. And like before, we don't have time to show you absolutely everything. So we'll go through and you'll have to trust me that these all worked. And again, we'll share the results, the full results with everybody later on when we publish this material. But the check solution to do our stability check, it passed. Our calibration curve looked really good. It was better than 0.998. Our ICV, or initial calibration verification, also looked really good. And then we had our CCB, which turned out very well. We did our duplicate portions that were plus or minus 20%. And now we can get on to some of the data that we'll. Okay, so if we take a quick look at the reference materials, for this one we used a NIST 1640A just to check the instrument, and you can see that all of the elements that we have listed here on the left-hand side, we were within plus or minus 10% for the percent recovery for that NIST 1640A. And also shown here is our CCV solution on the far right-hand column. So that's the check solution, the CCV, and that was at 1 ppm. And we were within plus or minus 10% for all of those elements. Okay, we're also showing the certified reference materials, which were the same as we used for the 4.7 method. So we can give a check and give a check, and we can move down to the Fortified method. And you can see here all of the elements that we looked at, they all passed for the 50 parts per billion spike. Okay, and then we have our Fortified analytical portions. So again, we used our beef jerky and our gummy bears, and everything was within the acceptable range of plus or minus 20%. So we can give check marks for both of those. And then another intelligent feature that I wanted to show you for the ICP-OES is our new star ranking system. So here you can see a little snippet of what the software actually looks like. And so this ICP-OES IntelliQuant data automatically produces a star ranking system for different wavelengths. And so this information enables you, the analysis, to see the best wavelengths in any wavelengths to avoid for your specific analysis, your specific method that you're running. So this is a brand new feature, and I believe that Agilent is the only one that has this smart star ranking system to let you and help you pick your different wavelengths. Okay, so here's some quantitative results for the food samples. And what I did here to kind of, again, reduce the size of the table is I added the 4.4 elements. So you've already seen the concentrations for a lot of the other elements. And these are the additional elements that 4.4 gave us. Okay, so I guess looking at this, nothing really jumps out to me as super surprising with the different elements that we're looking at here. Okay, so in summary, we showed software tools developed for ICP-MS and ICP-OES that use semi-quantitative data in several ways during the analysis of various food prepared and analyzed in accordance with the US FDA's 4.4 and 4.7 for food and related products. And in summary, again, I would hope that you enjoyed this presentation. And we here at Agilent and our friends at CEM are more than willing to help you with your food analysis in your different laboratories. And so just reach out to us and we'll see what we can do to help you out. Okay, so both of our instruments, the ICP-OES and the ICP-MS, are excellent choices for your food lab. They are both well suited to analyze food, and it just depends on what methods you need to run, which elements you need to run, what's your different lab environments. So feel free to reach out if you have any questions on either of these instruments or any of the data that we presented today. And with that, we'd like to thank you for listening to this talk today. And also, we'd like to thank all of our friends at CEM and Agilent for helping us with this study that we completed. So you've met Elaine and myself, but the other folks that we've worked with are Leanne, Macy, and Greg. And so a big thank you to all of them.